I am a marine biologist. I call myself an oceanographer at times, but I've also always been very interested in behavioral ecology and life history theory and fisheries, and sort of trying to blend those fields together. I've been teaching a course we had which was in general called aquatic ecology at the University of Bergen, which was a rare blend of freshwater ecology and marine biological oceanography. And I think that was a very nice, interesting course to bring limnology and oceanography together in a single course. Both parties, both limnologists and oceanographers learned a lot from that. I've taught also general ecology, the basic Bergen and Harper and Townsend big book, if you've ever been through that. And I've taught behavioral ecology, which is the Krebs and Davies, and the recent edition of Stuart West. It's a wonderful book also. It was a very interesting course. So that's my sort of core subjects. I also personally, I grew up in a fisheries community and a fisheries family actually. So when I was young, I used to go at sea and actually participate on fisheries every summer since I was 11 years old. And my brother is still a fisherman, so I do get that kind of impulses all the time. Also, I keep touch with what goes on in the fisheries communities in many ways. Okay, so that's and I'm now currently the leader of something that's called Theoretical Ecology Group, which might not mean a lot to you, but it's because we are actually having ornithologists. Ornithology is an important part of our research group at the moment. So that's sort of a compromise to cover all of our activities. And this is the web page. If you go to this link, you will find information about us, about people there, and you will find all the papers we publish. I would have published everything we've published actually back to the 80s or something. So that's so I encourage you to visit that page. And then sort of I was coming here with a little bit of an open agenda, trying to figure out what I will be talking about depending on what you are doing. And I think I tried to capture, I've sampled you a little bit. So I know that there is a diverse group here, and many of you do. Well, I guess all of you are somehow doing having Baltics in high up on your agenda. And since you are the future, I mean, since you will be responsible for the health and safety of the Baltic Sea in the future, I thought actually I'd try and give you and discuss a model that has been published only recently, a few months ago, that gave me an opportunity to read it, and I didn't participate in publishing it, but I'm deeply involved in sort of the background. I know the history of it, and I also understand it very well because of where I come from, and I think I spent some time on discussing that paper with you. It's not one of the three I put into the course schedule. So I submitted it, sent it to you. So maybe if you put it into the Dropbox folder, people can have a look at it. The first author is Swedish, Matilda Haraldsen, and she's a PhD student with our group in part actually, and the Danish group.
Is it the Haraldsen?
Yes.
Can I ask something just?
Yeah.
I don't want to go into the details, but what are the penguins doing on the picture? because these have something to do with Norway.
This picture was, as I said to somebody, this is an artistic picture. This is made by an artist who was paid for developing an artistic presentation of Glowback, which was a program that ended some years ago. I was involved in that program. So it's mainly, you know, it's really, it's only to, it's artistic. It's not a model. But art is our models of the world, aren't they? So actually, this is it is a model. Okay. So what? Yeah. So that brings me sort of to the things that I thought I'd do was these four elements that I thought. Because what I told you, what I was telling you about the things I had been doing, my background, changed again in the last 2 years or 3 years maybe. I actually become much more interested in ecological or ecosystem processes, ecosystem modeling, which is something new to me. And I got involved in the trait-based ecosystem modeling community somehow. And I thought I'd start by giving you a little bit of a touch of this, because that's what I'm doing currently. So but it's still a bit immature. You know, it takes some years until you can turn around and look back and see what was the important thing. So this is perhaps a little bit more chaotic than the rest. But so actually, I think maybe I'll skip that part. I'm bringing you straight to the sort of mechanisms part, just to get to these two final points. One which is sort of the main thing that I've done through my career is the optimality theory modeling. With this exercise that you will be given. And I think that should be the last part, so that we can do it for as long as you feel it's worthwhile trying, or leave whenever you like. But I want to make sure that we have enough time for the Baltic stuff, which is a little bit of a challenge to me, because I didn't really do it myself. But I have read it. So, and I have a presentation which I got from. I have all the slides and the background from it, so building up to it. So I think that could work. So that's my suggestion. If you have any comments on the agenda, I'm open. I mean, I could talk about fisheries-induced evolution, continuing the stuff. I could do some of that as well. I mean, that's part of this element.
I would actually be quite interested in the trait-based.
Ah.
Yeah, I was thinking the same.
Okay.
Maybe it's like 5 minutes.
I could do it really quick. It's like I could just like an overview.
That'd be great.
Yeah.
It would be great.
Okay, I'll do that. Just like a quick
talk. Up now.
No, yeah, I'll easily get caught in all kinds of digressions, but
yeah.
If I do that as if I had 15 minutes, I can try.
That'd be great. Yeah.
Okay. I need some water first. But otherwise, just don't worry. I mean, please stop me when I mean, I can skip a lot of stuff here, and we can spend the time needed on the important parts. And I'll try and look and see if when you get tired, I'll try and make a break or ask a question or something. Okay, so that's okay. Let's try and see how this works. Okay, so but before we start, I'll do something that I've tried to challenge my students sometimes. You know who this is. This is Steve Jobs, and he, I mean, youngsters know that he really kind of love this guy, and sometimes people are afraid of computers. Biologists. How many of you are biologists? Yeah, me too. Definitely a biologist. I didn't encounter computers like programming until I started my master thesis. So that's where I started doing it. It's possible even for biologists. It's something you can actually learn. It's not as hard as many people think. But he said something important here, I think, which is important. I said, yeah, that this applies to us as well. It's computers. We use them at work, but it's something else when you start building computer programs. It's something more. It structures your thoughts, your mind, the way you think. yeah, I mean, it's a tool for thinking. Huh? That's the key thing when you start doing building programs. And then he says something like, this is something everybody should learn to do at some point, a little bit, try it a little bit. And many of the things that I do and have done are just small lines of code, really, that actually you can learn so much from just playing with a few lines of code and putting your thoughts and ideas into those few lines of code and see what comes out of it. Sometimes you get, very often, maybe more frequently than you think, you will be surprised by what the computer tells you compared to what your initial ideas were. So that's yeah. So that's I like this particular. It's very relevant for biologists, I think. Then there's another thing that I just recently discovered here, and I'm a great fan of TAD. I guess most of you are also. And here is a short TAD's video, and I'm not going to show it to you or anything, but this is also, I think, a very nice thing. I like to poke youngsters, young people, students. So yeah, I see you have all these fancy mobile phones and all these fancy computers and everything, but you are consumers of the computers. Yeah? You're not using the computers to really express your ideas. It's a bit provocative, but I think you know what I mean. It's only when you use the computers for like writing a program or something to express yourself. And this is a wonderful thing that they do at the MIT Media Lab, is to develop simple programming tools, which you can learn kindergarten. The aim is to make kids in kindergarten build computer programs. And here it says, the punchline here is, if you cannot use the computer to express yourself, it's like being able to read, but not to write. I think that's a wonderful punchline, and it applies to a lot of people today. So that's just my sort of motivational pep talk for anybody who's on the brink to maybe try and build a computer code. So you go and watch this TED talk. Okay. Okay, let me do this speed talk on trait-based approaches.
Will we get your presentation as well?
Yeah, I can PDF it.
Great.
Okay, when you look into the ocean, it is amazingly diverse. There is all these forms of life that has all these sort of solutions that has worked in evolutionary time. They have survived since the beginning of time on Earth. and when we do modeling, we only collapse this whole diversity range into a few forms like diatoms and flagellates, and zooplankton, maybe 2 zooplanktons, and rarely we include any fish or anything. So this is a typical ecosystem model representing biology, this wide diversity in biological life that we have. Now, one way to, of course, this is bound to be wrong. I mean, this is everybody will realize. Yeah, this is the first question biologists normally. Yeah, diatoms and flagellates and one zooplankton group. And is that supposed to mean anything? Yeah, it doesn't really impress me much. So how can we try and change these models to represent some of the diversity that exists in nature? That's one of the essence in trait-based approaches to me. And I don't have an answer yet, but here is one suggestion that somebody mentioned this at lunch or something. But this is the slogan of some of my colleagues. I spent 1 year at MIT recently with Mick Follows and his group, and I'll show you. This is their way of doing that. Assuming there is a wide range of species everywhere, And then some of them, these species, have a success wherever they are. And this is Mick, and this is Stephanie. That's two of the people I worked with at MIT when I was there. And they picture it like this. You have, you can try and, instead of talking about species or functional groups, you can try and make it a continuum, like traits, specify organisms along one trait here and another trait there. And then you can say that there is some feasible combinations of these traits, and that could be anywhere in this space. And then you can sort of seed these organisms into an environment, and some of them will work and survive there, and others will not. And that's what you would, then you would have some sort of emergency in the structure of the ecosystem. You will have some successful species somewhere and others in other regions. An emergence biogeography, they call it. And this is what, when you're at the MIT, you can build models like this. This is a global, I just have to show you the video, if I can. This is on YouTube. And I think it's, well, you can't really see it very well, but it's, you can look at it. But this is how they visualize things, you know, in these places. They have these big labs, and this is one of the places at the MIT. This whole model covers a whole wall, and it's just keep going and spinning around and just showing up. But what you see here, these different colors are actually different types of phytoplankton. So they seed in something like, here it's just 78 species that have been just seeded into the ocean. And then somewhere where they are successful in particular regions and less successful in other regions. So here in the cold water, there's cold water adapted species, diatoms, and in the nutrient poor regions here, it's more like the small competitive nutrient efficient species that can live in there. So that's one of the reasons I went there. I mean, oh yeah, this is cool. I want to learn more about this. But even if these models are really seductive, you know, this is something you really, yeah, beautiful, emergent biogeometry. When you start to look into the details of them, they still have some weaknesses, and that's one of the core weaknesses I think of is that they are parameterized on a scale where you have is a typical assumption about that the key trade-off in these models are the trade-off between the half saturation coefficient and the Vmax. Some of you will recognize this is a Michaelis-Menten type language, where you have either you're a competitive and you have a low half saturation coefficient, you're able to compete for very low nutrient concentrations, or you have a high growth rate and you're able to compete at high nutrient concentrations. Very common assumption, goes back to Tillman's model, if you know about that. But these are all, I'd say this is wrong. if I be a little bit bold, I'd say there is no trade-off between these two parameters. It's very much assumed, and then this is scaled up to this global level. But what you really need to do to find what the trade-offs are between traits is you need to understand the processes at a deeper level. You need to know the physics of ecology. You need to know exactly what the processes are. This is a typical bulk parameter, just defined as the half of the maximum of the concentration of nutrients, where the particular species reaches half of its potential maximum growth rate. It has no biological meaning. There is no biological meaning. It's just a parameter. You can claim that you can measure it, and you can in experiments, of course. But still, it's not a real property of an organism. And this is something we, first my colleague Doug Oxness, he worked on this for a long time and developed a model that actually does fill that gap in that spatial temporal scale, of nutrient uptake. They solved an important problem in this paper, and then we reviewed the whole issue of nutrient uptake in microbes and in relation to this typical Michaelis-Menten framework. And I'm not going to into any detail of it, but my point is that the essence of trait-based modeling is to find those exact traits and how they trade off. It's very often simplified in such a matter that it almost becomes meaningless, like this Bmax half saturation coefficient trade-off. Okay, now there is another element of these models that hasn't been included. Of course, there's perhaps the most important one, cell size. These global models, none of them includes the true cell size, the size of the organism. Well, it's not true. There are some coming that do that now, but this is something that has been ignored for a long time. Just assumed to be sort of built into that half saturation coefficient, really. But this is, of course, in the aquatic systems, in pelagic aquatic systems, size is a very important trait. Very many processes are structured along size gradients and size, body size. Okay, so this is where we want to go somehow. You have to understand the scale of processes at a microbial level and then reach trade-offs among traits of real organisms, and then we can try and use that in global models to scale up and see what the consequences are. So that's one line of doing trait-based ecosystem models. The other one, which I'll talk actually is essential for that paper on the Baltic that I'll return to later, is this killing the winner principle. And that's essentially the way we like to think of it. We call it the killing the winner principle, because this was developed, something that has been promoted very much by Fredrik Tingstad at the University of Bergen. And essentially this is but historically I'd say this is a well-known principle in ecology. It's like the keystone predator type theory, for those of you like that. It's the Oksanen model. It's really a Swedish export product, this model also. So,
sounds very Finnish though. Yeah.
Lauri
Oksanen, yeah, he's originally Finnish, I guess. He's in Umeå. But the idea is that when you have two competitors that compete for the same resource, normally they cannot really coexist. Really, there's typically one that is just better than the other in competing for that single resource. But that improved competitiveness comes at a price, and that's typically something to do with a higher death rate or a higher susceptibility to predation or sometimes kind. So you can normally think of and Fredrik Tingstad has developed this mainly for bacteria. The susceptibility to viruses, that's related to a fast-growing microbe, while a slower-growing microbe would be a defense strategist that could defend itself but grows slower. So this is a principle that actually can generate stability and along a trait space here. So there's a gradient or a trait space here, and depending on the abundance of resources and the efficiency of the predators, you can have coexistence in this system. It's also a trait-based approach. And I'll show much more of that in when we go to the Baltic example. You see what I mean. Okay, there is another one. This is it's perhaps an ill-defined or a loose concept, the trait-based approach, but this is one that is something I don't know how important it is, but it's really interesting. It's really fun to think about. And I'll show you what I mean. It's a behavioral mechanism. And here is an example from, because this is something that has developed also mainly on land, although we have it in many systems in ocean and lakes as well. And I'll briefly mention what this is. This is a grasshopper. This is a spider. And on old grass fields, you can do exclosure. You can do some enclosure experiments where you can actually manipulate the system. And here they have manipulated the system by switching one type of predator with another. So there's two spiders. One spider is an ambush predator, and the other is more of a roaming, searching predator. And what happens here is that depending on which type of spider the grasshopper is exposed to, it switches its behavior. When you have a predator that sits and an ambush predator that just sit and wait for you, you can reduce your risk of predation by moving less. So they actually do that. They reduce their activity, their grazing behavior. While when you have something roaming around all the time, it doesn't really matter as much how much you move yourself. So the presence of these two predators changes the behavior of the herbivore, and that has implications on the structure of the plant community. Which plants are eaten by the grasshopper? So it's a bit, but it's not a numerical effect; it's a behavioral effect. So it's the same abundance of grasshoppers in the field, but they behave differently, which changes the plant community, and that actually turns out to have below ground effects as well. That means that it has biogeochemical effects. So that's an ecosystem effect because of behavioral responses within the system. And that has several names: trait-mediated indirect interactions or risk effects and whatever. And let me show you this particular famous example from, Brooks and Dodson showed in 1965 how important fish predation is in structuring the zooplankton communities. And Mark Oman showed in a very famous example how the presence of the presence of I had another slide, but here there's two stations. One is shallow, one is deep. In the shallow one there is no fish here, but there are still these ambush predators. And then the Pseudocalanus in this station performs no dial vertical migration, or even an inverse, no dial vertical migration. But here in this station where there is, here in this station where there is fish, they do a normal dial vertical migration, while here, where there is no fish in the shallow one, where there is only this invertebrate predator, they do an inverse vertical migration. So they have an opposite behavior in these two stations, which are actually in just kilometers away from each other. So the presence of fish affects the behavior of this small herbivore, because in this system it's subject to predation from an invertebrate predator, which do normal dial vertical migration. But here it's subject to predation from fish, where it will actually be eaten by that during daytime. So there's a behavioral switch in its behavior. There is a switch in its behavior depending on which predator is there. It's the same effect as we saw with the spiders and that. And this is an issue that I have been working with recently, and we've developed a model, a very, very complicated model, I should say. So I'm not going to say a lot about it, but it's taking this sort of typical model, and we solved the whole zooplankton community into a size-structured community where you have sort of many species approach. So some species can grow from small to large. And then the large zooplankton eat the smaller ones, and the larger ones produce eggs for the species that it's sort of representing. And then we have a fish, fish biomass in the model also that eats larger zooplankton more than smaller ones. So we have this kind of interaction where fish prefer the large ones, while the large zooplankton prefers the smaller zooplankton. And then you add the behavior. You have an optimal, an instantaneous trade-off between growth and mortality, both in the fish and in all of these zooplankton. So there's a very dynamic behavioral component in this model. Yeah, and on top of that, we also have an optimal foraging behavior in the fish compartment here. So this fish ignores smaller prey when it's not profitable to include it in the diet. And we identified when you have these type of, when you have a predator, they can eat different prey. These models run into some difficulties. You have to specify these preference functions that Stefan also mentioned, and what they are not, they're often specified by the modeler. It's something you choose rather randomly, and often the switching preference function is preferred because it stabilizes models. But that's during this process, we also wrote a paper about these things here, and one of the things that should be included in ecosystems models is perhaps a simple way where organisms choose prey to maximize their own success rather than following specific preference functions. I'll leave it with that because it takes too long to really go through it. So just to show you kind of pictures we get from this, we get for each size class you have a spatial behavior, a temporal vertical migration for the larger ones. The smaller ones tend to stay near the surface. Larger zooplankton come up at night and graze at smaller ones. There is a lot of things going on in that model. I'll spend the next few years and try and work on that model, I think. Okay, so that's sort of 3 elements of how to add traits into ecosystem models. I'll stop there. That's a quick tour through a wide round, I think.
Thanks.
If you have any comments or questions on that, maybe we should.
Just swallowed my water.
Yeah. I know this is I'm still confused myself in how all of these elements that are being called trait-based and that could be built into ecosystem models.
It's computation. It's very demanding. Also, it's done on the individual.
Yeah, we should look at the MIT model there, the Darwin model, as they like to call it. Then that's they can do it easily, and they can add thousands of species in there.
But it doesn't mean because they are from MIT and.
Yeah, of course.
Yeah.
I wouldn't be able to do that. But the other one, it's not really, but it's not spatial, so it's nothing where we put into like a 3D model. We have actually started to collapse it, make it simpler, because we can do that when we have a complex model. We can reduce the complexity, and if it doesn't really change the model, we've tried to sort of compress it into smaller boxes now.
Yeah.
Can you maybe briefly give some simple examples for traits and how to quantify them?
For the Darwin model, it's the Vmax and the half saturation coefficient, and then it's like the temperature optimum. So they have their mixed species along those 3, particularly along those 3 traits, they call it. So in the argument that I would put in here is that, okay, for a competitive ability of a phytoplankton, you would need the cell size. That's definitely, you need the cell size. and you need the ported density. How much of these uptake sites does it have on the surface? But in some sort of trade-off, and we don't really understand that trade-off very well. So that's two key traits that I would put into a phytoplankton model.
Mm-hmm.
And for the for that microbial model, that killing the winner model that Fred is working with, it's typical. If you grow fast, you have some. You're not having all the mechanisms there are also it's related to how intensively you defend yourself against viruses. So that's been a big discussion in the microbial literature. For the zooplankton that we worked on, it's of course the size there also, which size range these copepods are in, but and then we just added the plastic behavior, more like responsive behavior. I think that's feeding modes, whether they are filter feeding or cruising, for instance. That would define which type of prey they will be. Mm-hmm. They will switch from being filter feeders, for instance, or if you can call that some sort of suspension feeding, and switch to being a cruising predator if that's more profitable. Just simple algorithms that make the animals flexible in how they feed. Well, this switching models, switching preference functions that's been used now, they are essentially assuming that predators spare their prey, that they actually switch away from something that is profitable at low prey densities, which is not realistic. Do you need a break? No?
No.
Okay, so I'll slow down. I'll slow really like shift completely into something much more simple. And that's about the mechanics, ecological mechanics. And now I think you've seen some of the points that I'll try and make here. And that's, but here is a telling example. Here, assume you have made an experiment, measured how much a fish is feeding as a function of your prey concentration. And you would typically find data looking something like this. That there is low feeding rate, low prey concentration, and then it saturates at some point. So what do you do with this data? Well, what do you do with it? When you have done something like that, what do you do?
So I want to try to get a model.
You fit a curve.
Okay.
You fit a curve. That's what people do. So yeah, so you try a linear model. That doesn't work. I think you have to check your residuals or something. Students often do this. but then you do something like this, and then okay, yeah, you get. This is a model of the feeding of the fish as a function of prey concentration. Have you learned anything? No, I say no. But you can, and then it's tempting also to take this curve and use it in bigger models and use it for all sorts of things. This is saying that this is the relationship between the fish feeding and the prey concentration. Put it into any other model. Maybe you can find something that fits the data better, I guess, if you add parameters. But what if you try and think a little bit use theory a little bit first? And how many of you are familiar with the Holling disk equation? Only 2, 3. I'm deeply shocked. This should be in the sort of the basics of every ecologist and biologist, I think. What is it? The Holling-Disk equation has the history is that Holling in 1950s, he had a lot of students and he blindfolded them and he tied some discs on their hands and he let them move around blindfolded on the floor, and they put out some tiny items on the floor, and then the students counted how many encounters there were, how many times they encountered and were able to collect items on the floor. And then he developed this beautiful and simple model for the capture rate of organisms as a function of the density of the I can't remember what it was he put out on the floor. It was something. What? Anybody?
I said candy.
Candy, yeah. But anyway, and this is a model of exactly that, feeding rates of a predator as a function of prey density. And it assumes that the predator can only use its time for two things, either searching for prey or handling prey. And the time spent handling within a given time interval will be the number of prey it has encountered times the time it spends handling each prey. And the number of prey encountered will then be a function of search efficiency, clearance rate. This has attack rate. This has many names, but this is a crucial parameter. The search efficiency of the predator. And this is and the density of the prey and the time spent searching. Huh? This is all very simple and logical. There's another thing I always emphasize when you do these things, and give things dimensions, units and dimensions. That's the essence of modeling. So I'm used to think of aquatic pelagic systems. So there we use cubic meters per predator per second. That gives immediate meaning. Huh? That gives you a very clear meaning of what this parameter is. It's the volume searched by a single predator per unit of time. Perfect biological, physical meaning. And of course, units and dimensions must add up and match. Of course, they must be. You cannot add apples and pears in ecology. So then you can do something else. We have this, and then but we want an expression for the feeding rate, the prey, number of prey eaten per time available, and this you can actually simply derive from this assumption, and you end up with the Holling-Disk equation. This versions, the number of prey eaten per unit time is a function of the search efficiency, the prey density, handling time, the three elementary key parameters. Huh? I'll give you the PDF here. You can work it out yourself. Yeah, I'll return to this. Now let's return to this study we had here. Huh? We have made an experiment. Now, if you have the Holling-Disk equation in the back of your head as the theory of this process, then these data become very much more useful for you. Because if now you know exactly what the form of the theory of this process is. So you can fit the data using that form of the equation. Huh? So you get two parameters here, and not only that, you get an estimate of what is the handling time, and you get an estimate of what is the search efficiency, which relates to this figure here. If you think of this fish swimming, I mean, the volume searched by an individual fish depends on its prey detection distance, and it's so that there is sort of an area here that's scanned as it moves forward through the water. So you can formulate a model where you have image area here, or the search area, and the swimming speed determines a volume. And the handling time, you know that. So here, when we go back here, we actually have the estimated these parameters. And this is the image area, or this search area of the fish and the swimming speed. We're not able to separate the two of them in this here, but we have an estimate of the combined value of that and the handling time. This is the time spent then handling each particular prey item.
But isn't the efficiency not only depending on how much you filter? I mean, it depends if you're filter feeder or if you're raptorial. I mean, that's also tells about
This assumes raptorial feeding.
Okay.
That you lose some search time every time you encounter a prey and eat it. Yeah, so that's the basic holding this assumption, really. But this is the use of of formulating your theories and mechanisms and combining it with experiments. And from here you can, I mean, one of the key questions here now is how is this big A? V is essentially a behavioral parameter. It's something that the fish chooses. It can swim fast or slow. A is a function of many other environmental factors, particularly light. If you're a fish and you're in darkness, you see nothing. So that area will be very, very small. Huh? And then this feeding rate goes to 0.
But doesn't your efficiency also goes down as speed? If you
Yeah.
too fast, you miss out on.
That's of course, there are some experiments that show that. Yeah. Okay. Now, exactly the same theory can be applied to phytoplankton. And I've already mentioned this briefly, but if you think of a phytoplankton as an osmotroph that sort of absorbs nutrients, what really determines its uptake of nutrients is the density of these porters and the handling time in each of those porters. And so you have a substrate concentration and a diffusion process and a capture. So this is like a many-mouth animal in something. You can think of microbes as an animal with many, many mouths. And the clearance rate will depend on the number of mouths, the diffusion process, and the handling time of each porter. Yeah. So exactly the same model will work for this, except now you have the n here will be the number of mouths, but otherwise it's exactly the same. Now this is what I already talked a little bit about. I'm not spending so much. I think I won't spend a lot of time on it. Just saying that going into these details, actually, you can formulate an explicit mechanistic model of nutrient uptake where you have the cell radius, the number or density of uptake sites, diffusion, substrate concentration at the boundary, outside the boundary layer, and the handling time of each porter or each nutrient uptake process as your parameters. All of them have explicit meaning. So it's, although many of these you can go out and measure directly or model from physical processes.
The number of mouths, is it constant within the species, or is it
I don't know. You know. That's what I ask you.
I would say it varies depending on what is the substrate concentration. That's great from the point of view of microbes because it's own protein. So if you have less substrate, you can produce more transporters in your membrane. You absorb more than others. So
Yeah, no, I mean this is all very. I mean, there's a lot of papers coming out on just this issue at the moment, and what are the trade-offs exactly in this process when you're a competitive microbe, sort of versus Frede likes to call them a greedy microbe versus a fearful microbe. But what are the trade-offs? I mean, we don't really know. I mean, if somebody could help out on this is exactly a very vital area of research at the moment, actually. In ecological modeling context, it sort of links to the biochemistry, the really deep biochemistry of
especially
the organisms.
When the substrate is critical for energy production,
yeah,
and not only for growth, but for strictly producing energy to grow. So,
yeah, no, there's other elements with the growth of microbes. Yeah, let me skip this. Okay, that was an example of some mechanics. Now I want to have a herring story where we use exactly that mechanics to understand a bigger example of the ecology of herring. And I think I can do that in 10 minutes or something. Is it okay? Yeah. Fattening. Now we're talking about the big herring stocks, the not the small stocks you have outside here. It's this intensive, many million tons Norwegian spring spawning herring, which has this life cycle. So it goes to it overwinters along the coast in some fjords, and swims south along the coast, and spawns all the way down along the Norwegian coast, and then afterwards it swims into the Norwegian Sea, where it feeds and fattens over the summer. And then larvae drift off to Bergen, spend some early years here and go and join the parents at the parental stock after some years. That's the basic life cycle. They are conveyors of fat, an enormous amount of lipids, basically produced by phytoplankton, eaten by copepods, and then eaten by herring, and it's transported into the coast. And it's just a crazy amount of energy that is transported from one ecosystem to the other. We tried to find something that the amount of fat that is transported amounts to 10 times the total transportation of all of the salmon stocks in North America. It's 10 times as much as that. So it is we try to find some horizontal transports that are larger by any animal on Earth. And I think this is up there among the biggest ones. But that wasn't the point. The point here is, if you follow the lipid cycle of the herring, it looks something like that. They are decreasing their fat content over the year, and then in summer, just a few weeks in summer, that's when they gain all their lipid reserves. All that fat is consumed over a very short period, or gained over a very short period of time. So this is striking. This repeats every year.
Why is that?
Exactly. Why is that? That's what that was what was our question too. What goes on here? I'll try and explain what we thought was going on first. Yeah. So this is the average over a long time. Now, this herring feeds on Calanus phimosicus. This is also an animal that most marine biologists have a relationship to. So they overwinter in deep waters, very deep in the Norwegian Sea, comes up in early spring, and then develops a first cohort and builds up a lipid reserve at some point. Then they descend take their lipids reserves back with them. Aha, huh? Do you think the same thing that we thought? The herring could either be feeding on this generation, we thought taking the first copepods that come up, or they could be feeding on these guys here. They're only present for a few months in the summer anyway. So maybe there is some sort of match here that causes this lipid buildup in the herring. A natural idea of a hypothesis that we had. So to test this, we found the best data available from this weather station M. There was this is a boat that was present, although people are sampling copepods over the full year cycle twice. There's been one in 1949 and there's one in 1997. And they collected these data here, so continuously. You see here the first generation of colonists comes up, and then there is a large number of the adult stages, or the late copepodid stages, which are containing this big fat content in both years. You see it more. There are more of them here than here, but still you see the signal of the generations. So of course, yeah, this should be the reason why we would find these fattening of the herring. So putting it like this, putting those two time series together, and then we will see the abundance of the copepods over time. Yeah, and then we put the herring fattening on top of that. What do you reckon? Is it, it doesn't match, huh? It doesn't match. The fat, they build up their fat, and then the copepods come up. This is like 60 to 80% of the diet of the herring. It doesn't match. And yeah, but then what could be? Well, let's at least try and model this. What goes on? What happens? What's important for the feeding process? Huh? And I've primed you for this. You should be getting some sort of yeah, maybe type experience here now. Huh? Could it be?
But how is it with the production? Now you show a couple of abundance. No, that's not.
Yeah, no, that's okay. So you could argue that what happens here is that this is there's a big production going on here that's actually eaten by the copepods.
Probably it's too cold.
Yeah, but that's unlikely.
Yeah,
and because no, it's not the temperature, but it's because of the relative biomasses here. Although the herring is an enormous biomass, it only eats 24 million tons of calamus, and that's a tiny fraction of the annual production of something like that. So that's. I don't think that is actually. It's not that they're keeping them down. But what really determines the prey intake? Well, it's this magic equation again. Prey consumption, clearance rate, search efficiency, handling time. That's the two factors. And prey abundance. So we had the prey density. We know a bit about the handling time, and we can calculate search rate. And to get a daily prey consumption, we would have to integrate over 24 hours. And we know how, for this one, we could argue that there is a day-night cycle on the search efficiency of herring, how efficient they are feeding. Huh? And it would vary with the time of the year and the time within each day. For this one, we have data on how every day on how many colonists are available. And there are actually people who have been sitting watching herring and see how much time they spend handling a single prey. So you can find data on that. And then we're back to this. I already walked you through this model here. But the essential parameter here is this detection distance. Huh? How far away a herring detects a copepod is exactly what determines the volume search. That's the single most important parameter. And there is a detailed model developed for exactly that process, which involves eye sensitivity. it varies enormously among fishes. It's a trait. Some of the deep water fishes are extremely sensitive in their vision. The gobies that you have outside here, or something, that they need to protect themselves from daylight, so they're not as sensitive at all. And then there is a contrast of the prey. Some prey are like invisible because you can see through them. Huh? Others are very conspicuous. And then the size of the prey, how big they are in their image area, is a factor here. And the light. Now, if you do the experiments, you will see that the more light you put in, you will only increase the reaction distance to a certain level. So this is something that has this type of shape. So we use that model, and we parameterize this for herring as a function of light, but based on some old experiments that's been done. And then if you look across the time of the year, you will have variability in the day length, and you have a summer period here, which you have 24 hours of light up in around that station M, and the totally integrated amount of photons that comes to a surface will be something like that. So now we have all the ingredients, because we have the light enters in that detection distance. The detection distance enters the Holling disk equation, and we have the prey densities over time. So we can calculate. First, we can calculate the number of cubic meters a herring would search through when it swims through the water at a given depth, and that will be very much depending on the total irradiance. Then we can add how much energy it will consume. We can add that. That's just adding the. This is adding the Holling disk equation, integrating over the day, each day, and multiplying it with including the density of prey. So we know how much energy they would feed over the year. And notice how the prey density disappears. Yeah? You can see, okay, here there were very low prey densities at this point, but there's no, it doesn't look at all like the curve we had before, when we only look at the prey density. Because now light actually is a much more important variable in the search efficiency, in the encounter rate, than the prey density of copepods. Yeah, so now we can put, so this is what we had in for the prey density, and if we add this energy intake, average over the 2 years, it looks something like this. And that's more, I mean, to me that solves the problem in many ways. It's a sort of, look at how the food energy intake increases well in advance of the prey abundance because of that enormous increase in the photon flux in that period. Both the day length, total irradiance, everything increases in that period, and that suggests to me that, okay, that maybe there's some level here where herring, some level of energy intake, if that is at that level, then that's where they can start building their lipids. So they fill up, suddenly there is enough foraging efficiency at that particular time for them to forage efficiently. I mean, I think that is, I don't know, I think this is a sort of, to me it solves the problem. I don't know if it does for you.
But that doesn't, that solves the problem for the first part of the graph, but not for the later in here, because they would...
Here?
Yeah, there's a second...
this one?
Yeah.
This one?
That goes above.
Yeah, they are full. I mean,
they're already full.
They cannot really... So I don't know, but I mean, I think this is. I mean, you wouldn't think of this without kind of walking through that argument. That actually, what determines the food intake is not just the prey density, but also this, how much time they have available for prey search. Okay, that's my conclusion. In. I don't know if you buy it, or if you have any alternative hypothesis, I'd be happy to hear about them. No, I need a break. But I mean, if you have any questions, I mean, please. I mean, I like this example as a sort of a what can you use a mechanistic model for in some sense? It's a tool that can be useful actually to do sometimes. Let's try and think carefully through what is the process that you're actually thinking about, and put numbers to it.